Can microorganisms live in the upper atmosphere? Whether it be the troposphere, stratosphere, or even the mesosphere. That is what we'll be covering today. Biota of the upper atmosphere with Science Spotlight number 4. We're talking aerobiology and exobiology here. Bioaerosols across the troposphere, stratosphere, and even the mesosphere. So let's get started. The atmosphere is the largest connected environment on Earth, going all the way from ground level in the troposphere, up, miles and miles, kilometers and kilometers, until you reach the edge of space. Now, humans are certainly subjected to airborne effects from biological organisms, biological particles. It's very obvious if you ever have allergies, that's from pollens and spores, that'll affect people. And also, of course, the big one, communicable diseases, whether it's bacteria, viruses, they can pass through the air, certain airborne ones. So we know that there are biota in the air that can affect humans, that can be important. But microbiota certainly reside in the atmosphere. But there's a real open question whether microbiota, especially bacteria, can actually use the atmosphere as a habitat, and really how high up they can actually exist. And this question about what microbiota can exist in the atmosphere, how high up, etc., that has important impacts on humans, like human health, of course, agriculture, weather, ecology, and space technology. So this is not a purely speculative question. Let's now turn to some specifics, especially the term bioaerosols, which really goes into what we're talking about with biota, microbes in the atmosphere. So bioaerosols are atmospheric particles released from the biosphere. So that's living or dead organisms, that's dispersal units, so think pollen or spores, and then also can be fragments. And they can range from a variety of sizes. A good review of this is Bioaerosols in the Earth System, Climate, Health, and Ecosystems Interactions by Freilich, Nowicki et al. There's a whole range that of bioaerosols that are in the same realm of actual aerosols, but you can have things as small as proteins and viruses, which are on the ultra fine level. You can have bacteria on the fine level, and then spores and pollen would be larger on the coarse level. And of course, there's more than just biological materials when it comes to aerosols, but bioaerosols are what we're interested in here. Now, where they actually come from, the source, the abundance, and what they actually are made up of, and how they affect the environment, how they affect the atmosphere, it's not really well understood. And looking at bioaerosols can be broadly looked into 3 areas of research. Identifying and characterizing them, that's one. Second one is how they're transported in the atmosphere and how they transform while they're in the atmosphere. And 3, sort of the ecosystem interactions of bioaerosols. And now, there's a lot of ways you can actually identify bioaerosols. And the big one now is going to be with next generation sequencing, sort of being the gold standard, actually being able to sequence genetic data. Now, an important thing, and we know that organisms and dispersal units and organic matter is in the air. In fact, it turns out they do a lot of things. They can be dispersals for reproductive units, so pollen and spores are specifically meant to be carried on the wind, certain kinds. Microorganisms can be actually nuclei for cloud droplets and ice crystals and precipitation, so they can affect precipitation. And it turns out in most terrestrial environments, bioaerosols constitute a large fraction of the atmospheric aerosol load. And unsurprisingly, they're mostly found in the lower part of the atmosphere, but not a lot has been done higher up. So it's hard to tell how high they go. And they have all sorts of infectious, allergenic, and toxic effects that can have, especially at ground level, especially at low levels. Now, a specific thing when you're actually detecting these bioaerosols really goes into it, especially in contrast to sequencing, is you need to employ special methods to tell if biological samples are living or not if you actually care about whether they're surviving up there. Because you can sequence DNA, but it doesn't tell you whether that DNA was from dead or alive organisms. So you need to make sure, if you're interested in questions like that, to have those methods. A great quote kind of summarizing this, again, comes from this paper that tries to get at our answering, well, are there microbes in the high atmosphere? Are they living? Are they doing things? The quote is, however, although the atmosphere is intensively discussed as a possible habitat, hardly any studies exist that reveal the metabolic activity of microorganisms during their residence time in the atmosphere. So that's still kind of an unknown. So now let's turn our attention to specifically how we detect these organisms. So there's two kinds of ways to detect bioaerosols, whether living or not, whether just dispersal units or specific organisms like single cell bacteria. It can be online or offline. And a great review of these methods are Bioaerosols in the Atmosphere, our comprehensive review on detection methods, concentration influencing factors from Huang et al. There's online versus offline. Online relies on manual sampling and identification, where offline detects them atmospheric particles directly. And you can have passive versus active. Active or passive, you just put something up there like a petri dish or a filter, where active you need something to actually draw in the air actively, like a pump, etc. And great figures from this review show different sampling methods, from filtration to impingers and impactors. And then on the other side of sampling, you need to have some sort of analysis. There'll be different analysis platforms, whether it's simple as culturing or microscopy, or sort of molecular methods like DNA sequencing, qPCR, or even flow cytometry. And then, so those are sort of the offline, where the online is directly detecting things with like lasers, LiDAR, spectrometry, etc. And really what you actually do, how you actually sample these bioaerosols, really depends on what your research objectives are. You know, if you want to see if they're surviving, you might need to collect them and try to grow them in a culture, or run some sort of assay, see if they're living or not. And again, some methods differentiate between dead and alive, so whether that's a question you want to answer will depend on how you sample. And there are different factors that will influence bioaerosols. It's found in the table in this review, but I have it summarized here. So temperature, humidity, wind, particulate matter, solar radiation, precipitation, and different gases all affect bioaerosols. So you need to take that into account when you're trying to detect this for research. And here are just bigger pictures of those images. On the sampling side, we have impingers, filtration, condensation, cyclones, impactors, and electrostatic methods, where the analysis is cultures, fluorescent microscopy, qPCR, DNA sequencing, and flow cytometry. And then those online methods, it's laser-induced fluorescence, or LIF, LIF LiDAR, so a combination of those two, mass spectrometry, and Raman spectroscopy. All right, let's turn to the bigger question. Okay, there's bioaerosols, we can detect them with a variety of methods, but is the atmosphere an actual habitat? So looking at this paper, trying to answer the question, 2014 from Lapin et al., the atmosphere, a transport medium or an active microbial ecosystem. So they look through this, some of these big questions. So the atmosphere microbiome really does mimic the surface ecosystem. So what we found from the surface, so the actual microbes from the surface, they go up to the atmosphere. That's where the microbiome gets its components, from the surface. But there's a lot of cycling, because the air doesn't stay the same. The microbes get put up into the atmosphere, they get deposited down, so there's a lot of cycling. And metagenomics has really been a game changer in this aerobiology, being able to detect what's there, not just cultural species, but all species of microbes, particularly there. But there are three big questions that this paper brings up that really haven't been answered yet, that are ongoing. So are there residential microbial populations in the atmosphere? We know that they're there, but are there actually resident ones that sort of have them, the atmosphere as a habitat? And can microbes really survive in the atmosphere, especially the high atmosphere? We know that they're up there, maybe some of them survive, but can enough survive and actually be selective against pressures up there to evolve and not just survive a little bit of time, but actually be adapting to it? And then are microbes active in the atmosphere? Maybe they survive, but they're just dormant. Can they actually be active and catalyze things and grow and reproduce up there? And so those are the outstanding questions. And of course, there's a lot of challenges you have to deal with when you're actually sampling contamination. You have to make sure you're just sampling what's up there and not, microbes from the ground or from your equipment. There's not a lot of microbes up there. There's low biomass, so you have to figure out how to sample enough that you can actually answer these questions. And there's a lack of standardized sampling, so it's hard to look across studies if we don't have a standardization. So those are some open questions. Let's get more into some details of where you think of microbes in the atmosphere. How could they even possibly survive? Just air. Well, there's two particular places where they maybe could have a real niche, could have a real habitat. Clouds and dust. The first one, clouds, from this Amato et al. Active microorganisms thrive among extremely diverse communities in cloud water. It turns out microbes are in cloud. In fact, they can form nuclei for ice formation, but they looked at microbial communities in cloud water from atmospheric station. They did high throughput sequencing of DNA and RNA, so you can identify active species, not just the DNA of what's there, but RNA, like they're actually metabolizing, they're actually producing proteins, etc. And they found 30 different prokaryotic phyla and 12 different eukaryotic phyla, mostly fungi. And some were capable of maintaining metabolic activity in the cloud. And that was based on the abundance of both DNA and RNA. And some probable bacterial inhabitants of clouds are alpha and gamma proteobacteria. But, and then next one here about dust, this paper from Alice Mall et al., functional metagenomic analysis of dust-associated microbiomes above the Red Sea. Looked at a functional metagenomics of samples from a global dust belt, looking at 10 metagenomes, especially looking to get like sort of a gene list. And they found that, yeah, there was actinobacteria, protobacteria, firmicutes with bacteria, and they were able to get different genes. And it turns out that no matter where they got it from, whether it's bacteria, archaea, or even eukaryotes, that there were coding for proteins involved in the repair of UV-induced DNA damage. But the real question, even though we found them in clouds and dust, and they seem to be active in some ways and against atmospheric stressors and then towards sort of actual activity, metabolic and reproductive activity, the question of what they do really remains to be answered. Okay, they're doing something up there. They seem to be surviving, but how much are they actually doing? And that's a bit of the unknown. Now, we've mostly been talking about just generally organisms, microorganisms, bacteria in the atmosphere. And a lot of this is at very low level or ground level or just in the troposphere. That's where like most weather is, where most clouds are, where most samples are. But are there microbes above the troposphere? Well, a fascinating article from 1978, Upper Boundary of the Biosphere. This is a Russian one. They actually use meteorological rockets fitted with specially designed analyzers to collect microbiological samples in the mesosphere. So that's an altitude 48 to 77 kilometers. They shot it up with a rocket after sterilizing it, and they actually did find species. They had these sort of plates, and they were able to get things to grow on them after shooting them up there. They actually had 2 bacteria species and 4 microscopic fungal species isolated. Micrococcus albus and Mycobacterium luteum were the bacteria, and then the fungi were Sercanella muscae, Aspergillus niger, Penicillium notatum, and Papillospora anomala. So there were organisms they could get to grow collected from the mesosphere. Now, lower down in the stratosphere, we have a more recent article in 2018 from NASA, this Smith et al. Paper, of actually taking this collector that could be connected into the window of a craft, and they actually ran this, looking up to 13.7 kilometers, so that's the lower stratosphere, and they got bioaerosols. They captured them on DNA-treated gelatinous filters inside a Cascade air sampler, and they were able to do studies. And this was nice because it was fairly inexpensive, flexible to connect to airplane flights, and could be removed, periodically cleaned, and get DNA out. And so it showed that there were bacteria in the atmosphere of up to 12 kilometers and were relatively homogeneously distributed. So there is collected survival of organisms of bioaerosols above the troposphere. The only one that I've seen in the mesosphere is this rocket one. There's some in the stratosphere. Here's a airplane one, but that doesn't end there. Helium balloons or weather balloons are another one that's used in the stratosphere. So abundance and survival of microbial aerosols in the troposphere and stratosphere from Bryan et al. Showed weather balloons. They put payloads to sample microbial cells and dust at up to 38 kilometers. So that's again in the stratosphere. And the concentration of microbial cells decreased with increasing altitude, as you expect, but they were able to look at cells under microscopy. They detected ATP, and they cultured. And they actually got 15 isolates out. We were able to actually culture up to the stratosphere. They found actinobacteria, Firmicutes, protobacteria, and they actually took some of these organisms, grew them, in culture, and then subjected them to stressors that you would find in the stratosphere, like desiccation and UV radiation. They found tolerance to that. So it looks like these organisms, you got living, bacteria from the stratosphere and showed that they were resistant to stressors from the stratosphere. Though, one big note is that if they get up to the stratosphere, it takes a long time for them to get back down through these Brewer-Dobson circulation cycles of like 3 to 7 years. So it's very unlikely that they survive long enough to make it back down alive, unless there's a faster cycling that they get through. And then a final overview, especially of the stratosphere, when it comes to living organisms, microbial life, is this review by Desharma et al. In 2020. A great review of studies of Earth's atmosphere, especially the stratosphere, especially for astrobiology. And an important point the paper brings up, that elevation is a limit to life that has not been fully explored. And we can see how talk about high atmospheric biota gets into something akin to astrobiology or exobiology. And they looked at a variety of things, again, this is a review, but going over all these microbes that actually have been in studies isolated or tested in the stratosphere. And another question that comes up again, or an open question that this review states, is that whether any of these microbes are metabolically active or reproduce is yet to really be determined. That's still an open question. That's still an uncertainty. Again, these organisms, these microbes, have to survive really extreme conditions like irradiation, desiccation, freeze-thaw cycles, low pressure, and lack of water and nutrients. So this is a real extreme environment. And that's why even people thinking about Mars, thinking about astrobiology, the stratosphere is actually a great place to test some of these theories about extreme living, Martian life, right here on our planet. And it really does show that several observations of the actual microbes from the stratosphere show that they are generally, if they're alive, they're generally dormant. They don't really have active metabolic activity. All right, so where do we go from here? What's some future work of looking at microbes in the high atmosphere? Well, a great quote. Again, we've seen that this is still an open question. Whether life can exist for any appreciable length of time in the stratosphere, let alone thrive, is really an open question. It seems not, but there is life up there. There is life that can survive, and it's really hard to detect. It's really hard to tell if what they're doing up there. So that's still the open question. Now, where do we go with this? Where does research probably will go? Well, first of all, I think, especially on the lower level, a lot of the sampling we saw from the beginning comes at very low levels, ground levels. There are some in planes and balloons, but not as much. I can imagine drones starting to become, they're already becoming a very important use in sampling. Like there's this article, Drones in Ecology. People have been able to do and sample very interesting things using drones lately. I can imagine sort of the low troposphere or upper troposphere, well, not upper troposphere, but above ground level, but below airplane level being a ripe area to sample with drones. If you get higher up here, I think the idea of this exobiology really starts to play in as people get more interested in space again, more interested in going to Mars, etc. The idea of exobiology will really intersect with aerial microbiology. All right, with that, here's all the reference from the papers I used. They're found in the description as well. So hopefully you found this topic of microbes in the upper atmosphere as interesting as I did. So thanks for joining me. This was supported by Transcend. If you have any scientific topics for these spotlights that you think I should highlight next, leave it in the comment below. I'd love to hear that. Until then, have a great day.